Our treatment, currently known as MC1101, was invented by Dr. George Cho, who led the discovery of timolol, the pioneering treatment for glaucoma. Dr. Cho recognized that there are many ways that people have looked at trying to treat dry AMD, and they all seem to treat the waste management or waste products that occur in the back of the eye. And he thought, well, if there are so many approaches or so many causes of these waste products, how do you get rid of them? And he looked at the cardiovascular model that says that blood brings nutrients to the tissues, takes waste away. And so he identified actually a portfolio of compounds that have the effects that you described. They primarily increase choroidal blood flow in the back of the eye. It has anti-inflammatory and antioxidant properties. What we are doing is using our topically administered, so it is a non-invasive approach. We are actually preventing the progression of the dry AMD. The Mystic Pharmaceutical Versadoser, they call it, was used in our first clinical trial.
Okay.
It is a desirable package in that it delivers a preservative-free drug. However, we determined that it was best for potential partners to determine what packaging they would prefer to have to deliver our drug. And since drug approval in the United States, a package comes along with the drug, so it's kind of like the drug decides. We didn't want to limit ourselves, and I do believe that Mystic's approach is a very good one, but that should be left to the marketing partner, not the drug development. As I was saying, also, our MACUClear's purpose is to stop the progression of the drug of dry AMD to wet AMD. And we do this by restoring blood flow, which has been reduced in the eye. This has been known for over 100 years, that choroidal blood flow or reduced choroidal blood flow has been associated with AMD. 50 years ago, we determined there was a study done that showed peripheral vasodilators actually improved the vision of AMD patients. Dr. Cho was aware of this concept, that by increasing blood flow, we could restore the normal mechanism of bringing blood to the tissues, take waste away, stabilize the Brooks membrane. Because if waste product accumulation occurs and it builds up in the Brooks membrane, it breaks down and allows choroidal neovascularization, that is the new blood vessels that are formed wherever in the body blood flow is insufficient. Like cracks in a sidewalk, these new blood vessels can get through it, go into the retina, break down and bleed. By restoring blood flow while the Brooks membrane is intact, we can actually keep that critical blood barrier in place and never allow the choroidal neovascularization to get into the retina. The fundamental difference, Liz, between dry AMD and wet AMD is the integrity of the Brooks membrane. Nobody goes to wet AMD with an intact Brooks membrane. They may get geographic atrophy. However, that can take a long time to occur. We are in a phase 3 clinical trial, a double-blinded study, where we are using visual function endpoints. We have been given a concession from FDA that our first trial, if it proves our novel endpoint, will be included in the total of 500 patients that we will need to submit a registration for NDA. And we're pleased to say that we're well into that study. We have an interim analysis allowed without penalty, which we expect to do about a year from now, and we'll see how the data looks. It is true that currently there are no approved treatments for dry AMD. at best, 25% of the population that has it may be able to reduce the progression through certain types of vitamins. However, that is not a valid solution for most of the folks that have it worldwide. Our treatment is in phase 3, as I mentioned. We expect that at best we will have an answer in this first phase of the phase 3 to give us an indication of its efficacy within a year.
Wow.
Given that that happens, we will then be recruiting, we will begin to recruit the remaining 440 or so patients that are needed for the registration study. That would require at least another year with the FDA will require at least 2 years of safety data. And even though our drug is known as a 505(b)(2), which means its safety and toxicity is known, which is a good thing actually from a drug development point of view, there are no toxicity or safety issues that are going to show up in this. It's more an issue of efficacy. So I'm guessing that through the NDA process and so on, that the earliest that we will have NDA approval is 3 years.
A technical question. This particular compound, is it a vasodilator?
Yes.
And how does it differ from other vasodilators out there, like alpha blockers and beta and nitrates and so on?
All good drugs are tissue specific. For example, if you take Benadryl to treat, it's an antihistamine. Well, we know that you feel kind of lethargic all over the place, even though you're just trying to stop some sniffles. Not all vasodilators are going to treat AMD. For example, we get this question often. How does it work? Well, it's an antihypertensive drug that treats blood pressure. How does that work? Well, it's a small molecule that generates nitric oxide in the endothelial tissues and softens it. That's a well-known vascular model. Well, do all antihypertensive work? No, this one happens to. Well, do all nitric oxide compounds work? No. I get asked, I will say to someone, do you know how nitroglycerin tablets work for people who have angina? Oh yeah, I think I know about that. So I have been forced to say, well, do you know how Viagra works? And it works similarly. Well, then why doesn't Viagra work as an as a dry AMD treatment? Because it has been studied. The answer is it's not specific to the tissues in the retina. And that's what Dr. Cho did in his discovery work. Knowing what he learned from his discovery of timolol, he knew that certain tissues in the retina respond to certain types of drugs. And he began his search focusing on those compounds. For example, our compound MC1101 has no effect on intraocular pressure, so he discarded that as a treatment for glaucoma. But he found that it was responsive to the retinal tissues on a sustained effect that Viagra doesn't do, but this drug does, and that's how it works.
Viagra may actually be deleterious. We know that it does cause changes in color perception.
There is that evidence, but more importantly, it does not have a sustainable effect on the tissues in the retina, and that's why it hasn't been used.
I think that's an important point, is drug specificity.
It took me a while to learn from Dr. Cho that subtlety, that it is tissue specific, because he did get it. We have over the last 5 years gotten inquiries as to why does it work or why doesn't this work, and it comes down to a good drug developer is looking for tissue specificity, so you don't have these systemic effects. A drug today, because of the Vioxx issue, has to go through a whole cardiac safety profile. We don't have to do that. Our drug is known, and It's delivered directly where the source is, and it's specific to those tissues. It has very little effect otherwise.
Yeah, I think that's a critical point, because I know in pharmacology we say all drugs have side effects, and its best known effect is actually its most prominent side effect, and all of the others are minor.
If
So
you took this drug, if you took the API, formulated as an antihypertensive, it does have in its highest dose side effects.
Right.
And those side effects are temporary. If you stop dosing, they go away. However, because the drug is delivered locally and in a level that you'd have to do, I think it's 1,000 times this dose before you'd have the same systemic effects.
Right.
So
I'm sure in your clinical trials you've measured plasma levels to see if there is drug absorption into the system, and those have been minimal.
There is none at our level, and in the level that it would be delivered orally, there are plenty of studies on that.
That's another important point. Yes. Okay. Well, this has been most informative, Philip. Thank you so much for keeping us in the loop, and we'll
And I will
keep
keep
the
you
good word
posted.
out.